itself has a hydrophobic end and a hydrophilic end. One end that is water-loving and one end that is water-hating. Those ends that are water-hating direct themselves towards each other. These chains here are water-hating. They're hydrophobic chains, and so they direct themselves inward. The heads of our phospholipid are water-loving, and then they direct themselves outward, if you will. And so this you can think of as outside the cell, and this here then is inside the cell. In addition to our phospholipid bilayer, we're going to find embedded in our membrane proteins. Sometimes the proteins will run through the membrane and serve as a channel to allow things to move across the membrane. Sometimes they serve as a receptor, allow things to bind to the cell and then transport them across the cell. You can imagine that this cellular membrane here is actually probably studded with all kinds of different receptors for the different things that the cell is going to encounter. Think about it. Anytime a virus lands on one of these cells to infect, it's got to be binding to a specific receptor. Anything that gets into the cell is likely binding to a receptor or protein that's going to facilitate its movement into the cell. We can also say for the moment that the electron transport chain, which I'll talk about when we get to metabolism, the proteins that are responsible for the production of ATP for energy production are also found embedded in the cellular membrane. Now, I mentioned to you that the cell membrane is involved in molecule movement. Regulating the types of movement across the membrane. Well, we can have simple diffusion that occurs. For instance, gases can diffuse across. Not too many things do. With regards to prokaryotes, simple diffusion is not a process that's used much, maybe just in the gases for the most part, simply because it requires a gradient that usually is not established to allow diffusion to occur across the membrane. Osmosis, again, osmosis is a type of diffusion, simple diffusion, but in this case it's water movement across the membrane. And then you have directed movement, or what we can call active transport. These two processes don't cost any energy. It's a gradient movement. This here process does cost energy. To move something across the membrane against a gradient is going to require some energy. So we can have active transport of things across the membrane, sugars, other molecules. But another thing that might be necessary is a process called efflux, where we pump things out of the cell as quickly as they come into the cell.
So those two ways are how molecules moving?
This is movement across the membrane, but it's requiring energy. So you've got two types of movement across the membrane requiring energy. Active transport is movement of things that the cell wants, glucose and other types of molecules. Efflux is a process by which the cell gets rid of things that it doesn't want that's moving into the cell. For instance, antibiotics. Efflux is a process by which the cell can get rid of antibiotics as they move into the cell. So it's pumping these things out of the cell, and in that way, maybe protecting the cell from the damage that the antibiotic might do.
Again, that's going to require energy though. So
Yes.
can we call efflux as an exocytosis?
No, I wouldn't call it an exocytosis. That's a slightly different process. This is actually a movement of molecules through a protein channel. So that's not exocytosis. So imagine exocytosis is a process by which the membrane fuses with a vesicle and stuff is released. In this case here, efflux, you've got a protein channel that is probably being opened to force this stuff out.
Is efflux the main way it gets rid of its waste?
No. That's not how it's getting rid of waste. It's getting rid of materials that it doesn't want that's moving into the cell, like antibiotics and things like that. Waste are things that have been digested, and that would be lost through exocytosis. So we're jumping ahead a little bit there. We're not there. But no, waste products are not going to be lost through efflux. Efflux is just a way of pumping out things that are moving into the cell that the cell doesn't want. You know? And it's going to require energy. It uses the proton motive force, which I haven't got into yet and I don't want to get into now. We will eventually here. But it requires energy to get rid of those things. And then one more type of movement across the membrane that I want to mention here, and that's secretion. Secretion now, these are things that the cell is producing. Efflux are things that are moving into the cell that the cell wants to get rid of. Secretion are things that the cell is producing that it is then releasing into the environment.
Is that like a diffusion, or is that need energy as well?
Secretion is going to require, probably going to require energy with the fusion of the vesicle to the cell membrane. So you can see the cell membrane very involved in what the cell actually does. Outside the cell membrane, we find a structure called the cell wall. The cell wall is a rigid structure now, and it's going to hold the cell together. If you remember in lab, we talked about protozoans not having a cell wall, and so they need some way to deal with water that's moving into the cell. We talked about they have that contractile vacuole that helps them pump water out of the cell so that they don't burst. Bacteria don't have to worry about that issue because of the cell wall. Water can move into the cell freely, but to a point. Once the cell has filled up with water, it's not going to burst because the cell wall will prevent that from happening. Just like we see with plants as well. They have a cell wall that will prevent them from bursting. It just allows the cell to take up water to a point. Protozoa and other organisms that don't have cell walls, they have to deal with water that might be moving into the cell. Contractile vacuole might be one of them. But bacteria don't have that issue. So the cell wall provides them with rigidity, holds the cell together. And then when we look at the cell wall, we can see that the cell wall is composed of a molecule called peptidoglycan that is uniquely bacterial. No other organisms have this molecule, peptidoglycan. Only bacteria have peptidoglycan. And then, how it is arranged allows us to split bacteria into two fundamental groups based on the amount of peptidoglycan that they have in their cell wall. When we look at the molecule itself, it really is a series of repeating protein units Not that this is all that important, I just want to show you the structure here. So you can see, this is what the peptidoglycan molecule is going to look like. And not that I would expect you to necessarily copy this for me or be able to reproduce this, I won't. But I want to point out something here. Some of the bacterial cells have their peptidoglycan molecules bound by a cross bridge like this. Those cells form very thick cell walls. In fact, their cell wall, if this is the cell membrane, can be as many as 30 layers thick, peptidoglycan layers with cross bridges between the molecules. So it forms a very tight structure. On the other hand, some cells have a very small amount of peptidoglycan. They have an inner membrane, and then they have 1 or 2 layers of peptidoglycan. Those cells that have a lot of peptidoglycan, we use a staining technique that was first established by a gentleman named Graham. And if they have a lot of peptidoglycan, they stain purple, and we call them gram-positive cells. Anytime I refer to a cell as being gram-positive, what I'm telling you is that cell has this large layer of peptidoglycan as its cell wall, maybe 30 layers thick. Those that don't stain purple, because they don't have a thick peptidoglycan layer, are identified as being gram-negative. But here's the thing, that's not the full story about gram-negative cells. Gram-negative cells actually have another membrane on the outside of this peptidoglycan layer. So, if we took this and blew it up, and why don't we just go ahead and blow up the whole structure here now, what we would see is that you would have